In the end of the Second World War, between 1944 and 1945, as the Allied forces closed in on Nazi Germany, during the winter the Germans executed a blockade that would cut off resources like food and fuel coming from farms. This resulted in the Dutch famine, affecting millions of people and killing thousands. Later on, after conducting some studies, it was found that the people whose mothers were pregnant with them during the famine were more likely to develop certain diseases. This likelihood for diseases is not seen as much on their siblings who were born after the famine. As it turns out from these studies, it was revealed molecular differences at the DNA level between the two. These differences were at an epigenetic level. But what is epigenetics? Well, epigenetics are described as the area of biology that studies changes made in the phenotype, but that are not visible at the DNA level. These changes come as a consequence of the environment around us. So, supposing you had a clone of yourself, meaning you and the clone had the exact same DNA, you could expose your clone through a very different lifestyle. For example, the clone could be exposed to trauma. And so, in the end, the clone will turn out very different from you. It would probably develop some mental illnesses. What happened here is that, despite your genetic code or your genome being the same, the way it was expressed was very different. DNA is expressed when it is read and transcribed into RNA, and then translated to proteins. So, when the DNA is getting expressed, the amounts of certain proteins that are getting produced can be different from your clone. A good analogy I found from the YouTube channel SciShow is that you can almost think of it as a paragraph, and you're able to read the paragraph in many different ways depending on the punctuation you give it. So ultimately, you can give several different meanings to the same phrases by changing the punctuation. But how does this happen to the DNA? What creates this punctuation? Well, the DNA has these chemical tags binded to it, and the set of all these tags in the DNA is what we call the epigenome. Some of these tags are methyl groups, which consist in carbon and hydrogen compounds, and they are the ones who are able to control the genome by saying what genes should or should not be expressed. So when a methyl group is suppressing a gene, when the DNA tries to create proteins, that gene will not be read, which means that you will not get the respective proteins from that gene. On the other hand, if the methyl group is activating that gene, it will be read and the respective proteins will be created. This process of expressing DNA with methyl groups is actually very important to create the different cells in your body. Initially the cells are all the same, but then they differentiate into several types of cells like heart cells, skin cells, blood cells, etc. In addition to this, we have histones, which are proteins to which the DNA will coil itself around. The chemical tags will also define how tightly the DNA will be around the histones, and genes that are more tightly coiled will be silenced or expressed less. So now we know what's giving the punctuation to our paragraph. Our paragraph is like the genome, and the punctuation is like the epigenome. It's the epigenome that tells the genome what to do. Okay, and here's the thing. Recent studies indicate that some of this epigenome is hereditary, meaning it will live on to future generations. So while most of the chemical tags get cleaned when you are creating the DNA for the future baby, some of them may stay behind. And this means that the decisions and experiences that you live now can have lasting consequences that might indirectly affect generations to come. So for example, in the fruit flies, phenotype changes that are induced by the environment can get a transmission across several generations. And similarly, the Dutch people during the famine, the ones who starved, could pass some of those epigenomes on to their kids. So recent studies have shown the influence of the environment in the development and physiology of animals. The interaction between the environment and our genes determines how individuals with the same or different genotypes will respond to a certain environmental change. Examples of the effect of the environment are not restricted to a few organisms either. For instance, temperature can be a major sex-determining factor in many reptiles. And in most turtle species, sex is determined or influenced by temperature during the egg incubation period. In this case, some methyl groups will help regulate the sex-determining genes. So basically, depending on the temperature, the turtle can be a female or a male. You see this again in Australian central bearded dragons, where the chromosomal sex determination, meaning the X and Y chromosomes, can get overridden by high temperatures, and thus producing female offspring instead of males. With the use of machine learning, scientists were also able to draw strong correlations between the mental group's data and your biological age. That is, how old your body seems, according to numerous factors like your diet, your sleeping habits, and so on. It was also found that the biological age was accelerated by a high body mass index and reduced with high levels of physical activity, low body mass index, and consumption of fish, fruits, and vegetables. But most interestingly, they found relations between diseases and your epigenome. As it turns out, outside influence can lead to your chemical tags silencing certain genes that would otherwise help to eliminate tumor cells. When your epigenome seems the most sensitive to outside changes is during the embryonic development stages in your early life. This means that as a child, your epigenome is more susceptible to the effects of the outside changes. If the environment can affect us so much, then surely our class will also influence our DNA expression. Lower and working class people usually have less access to resources, less access to good education, harder access to medical care, and even harder access to food and housing. Even middle class people have much less quality of life than the upper class, and it is known that less money and education leads to less opportunities. But it goes beyond that. Your class can affect you on a molecular level by affecting your epigenome. A new study from Northwestern University found that lower education and income have a likely increased risk for heart disease, diabetes, and many cancers. In this study, they discovered that lower socioeconomic status correlates to the certain levels of methyl groups across more than 1,500 genes. In addition to that, it is known that big hardships related to poverty can lead to psychological trauma and other mental illnesses. So, poverty can quite literally leave a mark on your genes. As we now know, DNA is not the only thing determining who we are. Epigenetics challenges the old ideas that human intelligence is only related to the DNA and that some people just have better genes. So the good news is that some of it might be reversible. The epigenome that you developed from your surroundings is not permanent and you can change it again. Of course, it's not so easy, and some of these chemical tags might stick and be passed on to your children, just like your ancestors might have passed on some chemical tags to you. However, it is possible to change a lot of your DNA expression with lifestyle changes. For example, experiments made with smokers show that smoking results in epigenetic changes. Smokers have less methyl groups in certain parts of the DNA in comparison to non-smokers. The difference is even bigger in heavy smokers or long-term smokers. However, after quitting, former smokers begin to get some of those methyl groups back in the genes where they had lost it. Eventually, they might even reach similar levels to non-smokers. So in the end, epigenetics brings both good and bad news.